Well, good morning. My name is Bob Berendt. I am in the Office of Lifecycle Drug Products, and I'm happy to be here to talk to you about some quality considerations to help you avoid common deficiencies that we see in transdermal ANDA applications. So here on this slide, I'm highlighting just a few of the quality topics shown in red that I'll be discussing today and their ties to clinical concerns. And due to time constraints, I'm trying to basically group some of these together into three different topics of, again, common deficiencies. One is the importance of controlling and understanding raw materials. Second is anticipating potential stability issues. And then the third is the importance of formulating and designing your TDS product with the end use and user in mind. So I'm talking about the importance of controlling raw materials because, in part because, as multiple speakers have discussed already today, transdermal and topical delivery systems do not need to be Q1 and Q2. And so that allows a lot of flexibility to applicants when they're formulating their product. And some of the quality concerns involve adhesives and control of the rheological properties and lot-to-lot variability therein. And another concern is with the impurity profiles associated with the adhesives as well as the other components, such as the films that are typically used for transdermal and topical delivery systems. And to illustrate the importance of adhesive rheological control, I have a chart here which shows a plot of viscosity on the y-axis, and then it's showing the adhesive supplier's acceptance criteria. So this is what the supplier of an adhesive, this particular adhesive, would allow as far as releasing it onto the market. This point represents a specific lot of adhesive that was used to manufacture a clinical batch submitted to the agency for approval of a TDS. And then post-approval, this is some data that was obtained for rheology or viscosity associated with the same grade of adhesive. And you can see that the following marketing, the lots of adhesive used don't have the same viscosity value as the clinical batch. And for this particular application, the applicant had noted some issues with patient complaints. And so just want to highlight the fact that the quality control of your raw materials really should be representative of the material used in the comparative clinical endpoint, comparative clinical study, and that inadequate control may lead to issues in future batches. I also want to highlight the importance of understanding the impurity profile of your adhesives. When we're reviewing the ANDA for a TDS, we're also reviewing any reference DMFs for the adhesives, including the impurities that are present in those adhesives. And what we often see in those DMFs is a long list of potential impurities, including monomers, initiators, and cross-linkers. But when we open up the ANDA and look at the same subject, we maybe see no impurities listed. And so this demonstrates a lack of communication between the supplier and the ANDA applicant, and it leads to additional information request letters and deficiency letters that could be avoided if that communication existed before submission of the ANDA. I also want to remind everyone that extractables and leachables are studies that are required for these particular dosage forms, and that involves looking for extractables that may come from the backing layer, any rate controlling membrane or scrim, the release liner, pouching stock, and ink. And I've seen multiple cases where ANDAs, while they're in the review cycle, they've determined that there is an unacceptable level of a particular leachable from a particular component, and they have to reformulate their product, changing the backing layer or some other, the release liner. And so it's very important to be upfront and qualify your suppliers ahead of time to avoid those type of issues. And also, I recommend that you follow 1663 and 1664 from USP closely when you submit your extractable and leachable studies. So just kind of to wrap up that topic, really important to choose your supplier wisely in order to understand how the critical material attributes of your material and how those will affect the critical quality attributes of the finished product. The second topic are stability issues. We see these fairly often in TDS products where upon aging, quality differences are observed. And what it always comes down to are those changes clinically meaningful? And the burden is on the applicant to demonstrate that they have sufficient understanding of the quality of their product and whether or not that will affect performance. And the example that I have of this is crystallization, where for most of these matrix transdermal systems, the intention is to have the drug dissolve completely in the adhesive matrix. But upon aging, what can happen is crystals of API will be observed, as shown here in the snowflake crystals. And this isn't just a theoretical concern. Relatively recently, we were reviewing a fentanyl transdermal application, and we had some drug product samples sent to the FDA labs for a completely different quality concern. And the lab found that 12 batches had crystals visible to the naked eye, as shown here with the red arrows. And those crystals were identified by the lab as being due to fentanyl. So the concern is, do those crystals affect drug delivery and adhesion? And again, the burden is on the applicant to demonstrate whether or not that is a concern. And this could have been avoided potentially if the applicant had designed its formulation properly to avoid crystallization in the beginning. So again, just highlighting the importance of understanding potential stability issues and the effects on shelf life and how quality changes may affect clinical performance. And then finally, I want to touch on the importance of understanding how your product is going to be used and patient use and safety issues associated with that. And the example that I have to highlight that is it's showing a laboratory scientist who is removing the release liner from a transdermal system, which a patient would do so in order to administer the system to their skin. And you can see that there's an opaque square layer that remains on the release liner, and that's not how this should go. So as just a schematic to explain what was seen in the video is that the red layer is the active portion of the TDS, and the blue layer at the bottom is the release liner that's removed by the patient prior to administration. So the release liner should be removed and thrown away in the trash, and then the patient should apply the entire system where the red layer is in contact with the skin delivering drug. But what was happening in the video is that the whole active part of the system was being thrown away. And so there would be no therapeutic benefit to the patient, and then also a safety issue associated with potentially improper disposal of the drug. And the safety issue that I've mentioned here brings up the importance of understanding residual drug. Because the agency understands that residual drug has a significant potential to impact quality, efficacy, and safety of a product, the residual drug guidance that's publicly available, it states that residual drug should not exceed that of similar FDA-approved products, and very importantly, that the amount of residual drug present in a system should be minimized consistent with the current state of technology. So as new penetration enhancers become more commonplace, the expectation is that applicants will start to look at those as potential ways to limit the amount of drug that's used in each of these systems. So the design and formulation of the product should facilitate correct product use by the patient and then also mitigate safety issues associated with the product. So in conclusion, I've highlighted some broad topics for your consideration to avoid common quality issues that really, they significantly impact our ability to get first cycle approvals for these particular dosage forms and result in multiple CR cycles typically. And I, again, want to highlight the fact that you as an applicant should really understand and fully characterize your system, particularly your clinical batches, the raw materials that go into those clinical batches, the manufacturing process used for those batches, and the stability, so that your clinical, the clinical studies that you submit to the agency are representative of the performance of the future commercial product. And so with that, I'd like to acknowledge my colleagues in OLDP, as well as the CDER Transdermal Working Group, and the FDA laboratories for their work in the slides that I showed. And I'll also point out that I've included some common deficiencies as reference at the end of my slide deck. And with that, I'd like to turn it over to Dr. Ying Fan to provide more of a clinical perspective.
Thank you, Bob, for the great talk. So my topic today, I will focus on the common deficiencies from clinical perspective. So it's a lot of things can cover for this topic. So I'm going to focus on today is the common deficiencies in the skin irritation, sensitization, and adhesion study. So this is a disclaimer. I think all of you should know very well by now, so I'm not going to go over this. So the outline for my talk today, first I will give an overview for the skin irritation, sensitization, and adhesion studies, what we call ISA study. And then I will talk about the common deficiencies identified in the review of the ISA studies. And finally, I will give a summary what is the take-home message that you can get from my talk, so you can submit a high quality of ANDA submission, so to get the first review cycle approval. So the overview for the ISA studies, the goal for conducting this study is to demonstrate the irritation sensitization potential and adhesion performance of the proposed TDS is not worse than the reference TDS. So the statistics method that we use is the non-inferiority test. So to evaluate the irritation and the sensitization potential, what we recommended in the current product-specific guidance is the combined irritation and sensitization study. It is a multi-center, evaluator blinded, randomized study, and it is composed with the 21 days of induction phase, 14 to 17 days of rest period, 5 days of challenge phase, and if needed, re-challenge phase will also be included in the study. So the treatment arms in this study is the to be marketed your generic proposed test products and the reference drug products. And for some transdermal system that have the safety concern to conduct a 21 days IS study, then we recommend that you also can use the vehicle TDS without the active component and also the positive control, which usually we recommend 0.1% sodium lauryl sulfate to conduct study. And to evaluate the adhesion performance, what we recommend in the current PSG product-specific guidance and the general adhesion guidance is adhesion alone study or the combined adhesion pharmacokinetics study. And it is a single dose, randomized, 2 treatment, 2 period crossover study. And the treatment arms is the to be marketed test products and the reference products. So the duration of the study is the maximum labeled duration of wear of the reference drug product. So this slide shows the easily correctable deficiencies or information requests that we send out for the ISA study in the ANDAs that we take action from 2015 to 2017. And why it is important for you is because for every ECD or IR that we send out during the review cycle, it will delay our decision or review for about a month. So it will be very valuable for you to know what is the reason that we send out ECD-IR and how to avoid this. And you can see from the table that the majority of the reason that we send out ECD and IR is the clarification and justification, which is about 76%. And I will talk more details in later of my slide. And second one is the formulation-related issue. And third is the missing case report form. And fourth is the missing data sets or data definition files. And you can see from this table, the total percentage is a little higher than 100% is because one ECD or IR could contain multiple reasons. So let's look at the first reason for ECD or IR for the clarification and justification. So if we see any TDS that was totally detached during the study, IS study, for example, and we will ask for you to provide the date and time when this TDS is completely detached. And if there's any patch was reapplied during that study, then we would like for you to provide the date and time when this new transdermal system was reapplied during the study. And second, if we see any inconsistent information, for example, between your study report and the case report form, or between the data sets and the data definition file, and we would like for you to provide explanation why it happened and which data that we should rely on during the review. And third, if we see the conflicting study results when multiple studies were conducted, for example, if you evaluate adhesion performance and you conducted multiple studies, and some results are passing the non-inferiority test, and some of them failed the non-inferiority test, then we would like for you to provide explanation how the overall conclusion was made. So when you conduct a study that is different from our guidance recommendation, then we would like for you to provide a justification why you're not using our guidance recommendation. So for example, if you use the different scales for the IS study or the adhesion study, and then we would like for you to convert the scales to our guidance recommendations and submit a study result. And the second example, that if there is any TDS that moved before reaching the excessive irritation score, then we would like for you to provide explanation when it happened. So the second category second reason that we send out ECD-IR is for the formulation-related issues. And we would like for you to provide the formulation and the manufacturing process for your proposed generic TDS and also vehicle TDS if it is used in the study. And if there's any inactive ingredient is different as a reference listed drug and is higher than the IRD database that we can allow the limit, then we would like for you to provide a justification why this inactive ingredient is safe to be used in your proposed generic transdermal system. And third, we would like for you to clarify whether the to-be marketed formulation was used in all of the studies that you submitted. So the third reason for the ECD and IR is for the missing case report forms. We would like for you to submit the case report forms for those subjects who were removed from the analysis population, demonstrate the protocol deviations or violations, experience serious adverse events or death. And we would like for you to submit a case report form for all the subjects, but at a minimum 10% that you have to submit. So the fourth one is for the missing data sets and the data definition files. And I provided some examples here. And we would like for you to submit a data set that in the SARS XPD format for the adverse events, concomitant medications, medical history, and for if there was any subjects was discontinued from the study, we would like for you to submit the data sets which include the reasons for the subject discontinued from the study. And also, we would like for you to submit the data definition files for all the variables in the data set. So here's the summary for my talk. I hope that right now you can understand the goal and the study design for the ISA studies. And please refer to the product-specific guidance and the general guidance on the assessment of adhesion when you design your study and conduct your study. And other approach may be acceptable, but will require the justification. So lastly, please provide the justification and clarifications or other information in your original submission. So here's all of my talk today, and thank you. And then I will hand over to Dr. Stella Grosser for give a talk for the common deficiencies from statistics perspective.
Hi, good morning. Much of what I'm going to say is really filling in some of, or what Dr. Fan already outlined, and I just want to sort of expand on a few points. Much of my introduction and my summary actually was stolen from her talk because I'm, again, just expanding on the clinical considerations and the statistical issues that sort of come up during review of the clinical part of these applications. So again, I'm going to talk about the statistical analysis of these ISA studies and just touch on some of the deficiencies that we see a lot, some of which you've already heard about in the previous talk. So the whole point of irritation, sensitization, and adhesion studies in the transdermal system, generic ANDAs, is to evaluate these potentials and the actual adhesive properties. We want to show that the performance of the test product is not worse or not too much worse than the RLD. This we refer to as non-inferiority testing. We're not looking for superiority as we are in new drugs, where it has to be better. We're not demonstrating efficacy at this point. We're just showing, we have a reference listed drug that holds on, that doesn't irritate, and we don't want to introduce something that will be considerably worse. One part is a combined irritation sensitization study, and then another part is either an adhesion study alone or as part of a combined PK and PK study measuring bioavailability in the blood of the products and evaluating adhesion at the same time, at the same sampling points. Irritation is evaluated at the time of each system change. And again, that will vary from product to product. It's evaluated on an 11-point scale. This is, as I understand it, standard in dermatology. It's a combination of a dermal response, which refers to the skin appearance, 0 to 7. And there's also an other effects scale that might measure edema, etc., and that's measured 0 to 3. And you can see these examples, in the product-specific guidance. And I had hoped to include these scales, but again, they're widely available. There are 2 different scales measured by the clinician, by the observer, and added together. So it ends up being 0 to 10. Adhesion is evaluated at each of multiple time points. And again, that if it's combined with pharmacokinetic sampling times, that's when adhesion will be evaluated. If it's a separate study, there'll be a well-designed description of when adhesion will be noted. And that's a 5-point scale ranging from basically no detachment, no lift off of from the skin, to 4, which is 0% adhered or falling off, completely detached, and everything in between. I don't have pictures. I was going to have a picture so you could see 0, you could see, 75 to 90%, what would be graded as a 1. But what happens, and I've seen this only recently, is that the detachment could be at the edges. There could be detachment sort of in the middle. It's a fairly complicated evaluation. And again, there should be a trained observer noting the scoring, doing the scoring. So the comparison is based on mean scores, averaging over all subjects, all times within each product. Irritation is measured or compared with a ratio of means. We want, again, in this non-inferiority setting, we want the ratio to be not very large. We don't want the irritation score for the test to be twice as much. For example, as the reference, if it's 1.1%, 1.1 times as much, 1.25 times as much, that's acceptable. But again, this is a statistical test, so we take the mean scores, we take the ratio, and then we calculate the confidence intervals around it, the standard deviations, and plus or minus whatever. Adhesion is evaluated as a difference of means. And again, we want that difference to be not very large. It could be a little bit worse, but not a lot. This is all detailed in the guidances. And so as Dr. Fan said, look at the guidance, study the guidance, RTFM, as we say in other businesses, and go from there. The information requests that we send out in the course of review are related to guidance deviations, to data set problems, and to some calculation problems. For example, it's sometimes misunderstood. We're measuring irritation or adhesion as per system, not per subject, because each subject usually gets one of each. For adhesion, since the guidance was released fairly recently, we say unequal sampling time is okay. So if you're measuring to capture the metabolic course, and you measure 3 hours, or you measure 1 hour, 2 hours, 3 hours, 6 hours, 9 hours, 15 hours, it's unequal sampling times, but we can adapt. We take a weighted mean of the adhesion scores. Data set examples, again, that Dr. Fan talked about. There's patch detachment. It can just be a mess, and we need to struggle through this. The cleaner your data are when they come in, the faster the review will go. There may be inconsistent information and conflicting study results. Calculation examples, we run into all sorts of problems. People, we've seen ANDAs where the evaluation is based on the medians. There are subtle problems associated with that, and the guidance really recommends mean scores. We explicitly say, but again, we see that people will include baseline values. We say, don't do that. Why not? Because almost certainly all baseline values for all products will be 0. It's not irritating when the system is put on. It's sticking, I hope, when the system is put on. So there won't be any differences. And it kind of biases everything towards 0. The mean scores towards 0 and the differences towards 0. So it's sort of not a fair comparison. And again, clarify. Our questions tend to be, please clarify what you did here. Please clarify what this means. Or justify, why did you do that? Why didn't you do what we recommended. And again, you don't have to do what we suggest in the guidance, but we need to know what your thinking is. So be deliberate about your analyses. Mistakes at this stage of the product development usually are not irreparable. You know, you gave us an answer, you gave us an application analysis based on medians. Well, just recalculate. Let's look at the means. It's not irreparable, but it's inefficient, right? Because we have to go back and forth, and it could be misleading. Well, after you compare means, the adhesive properties might be a lot worse, but the median might be the same. And so again, follow the guidance, justify other approaches, understand the purposes of these studies, the study designs, the analyses, and communicate. Thank you very much. And I guess we're having a panel discussion.
We are having a
Yes.
panel discussion. So you pick
Yeah.
whichever seat is the most
Right.
appealing. I'm going to ask Tanaz to come back up and join us, please.
I'm also going to invite my colleague, Somesh Chattopadhyay, who is a reviewer and a little bit more in the weeds and can answer
Oh, wonderful.
possibly questions. I hope that's okay.
Okay, that'd be great.